Okay, I think we are ready to go. Happy Friday. So last chunk of material, the exam is set to open in an hour. So this exam, although we are right at the end of midterm week, this exam will not be included in your midterm grade because it's not. So there you go. Uh, all of that, I mean, it's open till Monday of next week, so it technically can't really count. So, that stuff, I don't know when you will see that stuff, but those grades have been submitted. They got submitted early this morning. So you should probably be able to see what's up with that. Maybe right now, maybe not. I don't know. I don't see those things. I don't know how you see those things. So, this last bit kind of wraps it up together and we start thinking about different techniques with genetics to think about ways that we may be able to manipulate things, techniques that we can use to screen DNA, thinking for like maybe paternity tests or something like that. There's all sorts of different applications. So I know we talked before a little bit about polymerase chain reactions and ultimately using this technology and using enzymes from microbes that are high heat stable for our own gains. This gives us the opportunity to take a strand, not like an entire chromosome, but a gene or maybe a handful of genes of interest, to use heat to unzip the DNA, and then the polymerase links up and new nucleotides get laid down, and it's like exponential growth. You get massive amounts of product in a short amount of time. So our thermocycler that we see here, we're not seeing like the bit right here where you would have your samples, but the heat and cool very rapidly, allowing for countless, not countless, but multiple rounds of replication to give you enough quantity of DNA that you can actually do some science with. Having one single piece of DNA isn't enough. We need to ramp that up. So oftentimes we think about trying to cultivate organisms, trying to grow them and extract the DNA, but we can flip it around for organisms that are maybe difficult to cultivate. So this image is some chlamydia, and I see this picture, I see these cells, and I know these, they call them McCoy cells, I believe is what these cells that are infected, I'm pretty sure that's it. These are cells that are infected with it. Chlamydia is an obligate intracellular bacterium. It has to be in a cell in order for that process life cycle to continue. So you can't just grow it on a petri dish. You have to have living, viable cells in order for that to occur. So there are other opportunities where we can do a different type of PCR where we're not trying to extract all of this DNA from a culture that we've grown up. We could take a sample, a specimen from the patient, and apply it to a PCR reaction and amplify what we have in that, eliminating the need for cultivation. And that sets the stage for dramatically increasing the time that we can diagnose, or decreasing the time, excuse me, that we could diagnose those situations, not needing any of that cultivation. So there's lots of different applications with PCR and different techniques that we may think about. We'll talk a little bit about some other stuff as we progress along. So when we think about dealing with molecular biology, we oftentimes think about taking a gene and transplanting it into something else, if you will. We call this recombinant DNA technology. And when we have made that transplant, it's a recombinant, all right? And now we've got some other gene that's been transplanted into an organism. That organism can grow up. And if we got all of this stuff in there properly, it can grow up and it will produce whatever specific product that gene was coding for in the first place. So here we've got an example with a human cell pulling the gene out and putting it into the plasmid. This technically is not exactly what's happening because we can't just take the gene out and put it into this plasmid. Why? What's up in a human chromosome that we don't see? Okay, circular and linear, but there's different stuff. So what do we have in those linear chromosomes that we don't see in the circular chromosomes? You remember this stuff? Not the telomeres, but telomeres are in eukaryotic DNA. That is true. What do we do if we're thinking about, we talked about this before with like making proteins, that messenger RNA in a eukaryote, there's other stuff going on in there, right? We have to process that. Introns and exons, right? Those introns are found, the in-between stuff. The introns are found in eukaryotic chromosomes, kind of like little spacers in between some of the details. And we have to chop that stuff out. So if we were to take DNA straight out of a human and try to put it into a prokaryote, it's not going to work because there's extra information in there that's not what we want. So you have to do some processing beforehand. And we'll see a little of that in a bit, but I feel like we're skipping it over right here, and I think it's important. This gene, before we are able to transplant it, what we need is the messenger RNA, and we need the messenger RNA to already be processed, all of the introns chopped out. So now we've got a piece of messenger RNA that in this case is insulin. Let's build insulin. And we take that messenger RNA and we expose it to an enzyme called reverse transcriptase. HIV has reverse transcriptase to take its RNA and turn it into DNA. That's what it does. So RNA, messenger RNA, exposed to reverse transcriptase, makes a copy of DNA. And this DNA now has all of the garbage, if you will, chopped out, and you get a double-stranded molecule, and we call that cDNA, or complementary DNA. It's DNA that's been generated from the mRNA molecule. So it's got all of the stuff chopped out that we don't want. And now we've got this gene that is DNA that doesn't have any of that information. So the extra step is super important if you're thinking going from some eukaryote into a prokaryote. We could forget about the prokaryotes and we could use a yeast cell. Yeast cells are eukaryotic. We're eukaryotic. We could take a gene out of our nucleus and pop it into a yeast cell, and it's got all of the machinery needed to deal with the introns, and you wouldn't need that extra step. But it's a lot easier to work with some bacteria and do that process, even if it's an extra step, all right? So then we use, after we've got this gene out, and technically to get these things out, we use restriction enzymes, molecular scissors, that cut DNA specifically. And we cut the gene that we want with an enzyme, and we use the exact same enzyme to cut the plasmid. So the cuts, it's like a puzzle piece. Now we're going to fit perfectly, all right? Don't force it. So the cutting that we see here is the result of those restriction enzymes. And there's different ones, we'll see that in just a second, that make differing types of cuts. So they merge together. Now we've got this recombinant plasmid with human DNA, and we insert it into a bacterium. There are a couple of different ways that that may happen. And once that's put in there and we grow these bacteria up, if all of this happened properly, all of these arrows, it's not flagella on these are little arrows indicating that these bacteria are secreting, in this case, insulin out into the media. And we would collect that, harvest it, package it up, and sell it to you. All right. Big business. So this understanding that we can replicate DNA with PCR, that we've got molecule enzymes that will cut these molecules into specific bits, opens up the potential for all sorts of genetic engineering or bioengineering, molecular biology, all right? Producing basically anything you could imagine, all right? So this is an example of a couple of different enzymes that make these cuts. We call them restriction enzymes. They're also called endonucleases. Doesn't matter, all right? The restriction enzymes, like I said, like some molecular scissors, make specific cuts, all right? And I had a student many semesters back. I've seen students wheeling around backpacks, you know, all sorts. They've gotten multiple books. So it's like they're wheeling luggage, right? Is what it looked like to me. I'm like, geez, that's too many books, right? This girl rolls in with this huge thing that she's wheeling in. I'm like, man, what's up? Scrapbook and stuff, right? Then I'm like, well, why the hell are you bringing that to class, right? But you never know, right? When you got a scrapbook, you got a scrapbook, right? So she's got all this stuff, and I'm kind of curious. I'm like, what are in all of these drawers? One drawer was full of scissors, and that's the idea that I'm not a scrapbooker, all right? Thankfully, all right? But I have seen people that are, and I know now there are more scissors than what I think of as a regular pair of scissors that's just going to make a nice straight cut. Pretty straight cut, you'd hope, if you know how to work a pair of scissors, all right? But with scrapbooks, there are scissors that make all sorts of pretty little cuts. All of those different pairs of scissors, we could think of like a different type of restriction enzyme. I'm going to cut like this, or I'll cut like that, whatever it might be. And they recognize specific nucleotides, all right, that we see this specific arrangement and we make a cut, all right? In a bacterium, this may be important if it's pulling in DNA from the environment, or potentially a virus tries to infect it, and now we've got this new DNA pumped into my cytoplasm. I've got all of these enzymes, and we just want to chop them up because we don't want that DNA causing harm, right? We'd like to recycle these bits if we're a bacterium, right? So that's kind of the idea of why they harbor these enzymes, is to degrade that DNA that they may be bringing in. And then once we've chopped it up, we could continue to degrade it and use the individual nucleotides as building blocks. But we could take those enzymes, knowing how they cut, and make these specific cuts, all right? They can make cuts that are like the fancy scissors that give you some sticky ends. So you see a little bit of overhang on both sides. Sticky ends, when we're thinking about trying to get stuff incorporated, work a little bit better because they match up like a puzzle piece, all right? There are other scissors that are like what I think about, a normal pair of scissors, just a straight cut, all right? These cut directly through there. Then you get two little chunks. We call these blunt ends. There's no overhangs, all right? And the issue with this, using something that makes a blunt cut, how do you know if it goes in this way or if it flips around or upside down? There are multiple potentials for that to not be inserted properly. So it's a little bit safer working with this to kind of ensure that you're in the right orientation. You can still do this with the blunt ends, but you might have stuff that ends up inserted incorrectly. We can test for that, all right? So we make these cuts, gene of interest, our plasmid, and now they fit together like a puzzle piece, and there you go. All right. So this is a plasmid with kind of numbers around it showing us there's all sorts of different regions within this plasmid that we may potentially be able to utilize. We're going to see some more of this in a second. But what we're looking at right now, this little piece of the entire plasmid, there are regions within here, all right, the colored regions. These are all areas, and each one of these is a different type of restriction enzyme. So small bits of that DNA that can be recognized and recognized slightly differently. This is where we make the cuts, all right? This is where our enzymes do their job. And it opens up that plasmid with the sticky ends at various nucleotides based off of the respective enzyme that's utilized, all right? This other bit up here, LAC-Z, this is going to be a gene coding for the utilization of lactose. That's going to be something that's going to be a marker for us. So we know, did it work or did it not? All right. There's some other stuff as well. Oftentimes with some of these plasmids, there's an antibiotic resistance gene that's in there. And if everything got in there properly, that antibiotic resistance gene will be active. You will grow these bacteria on a plate that contains antibiotics. So anybody that didn't get the stuff in there like it should is going to die. And if it did happen, they will grow. All right. You also, with that lactose, you can have lactose incorporated into the media as well. And so now it's growing. The plasmid got in there. But if the gene got in there, the lactose gene will be active and it will light up. All right. So you can see it growing and then see a color change to verify not only did I get my plasmid inserted, but my gene of interest also is in there. All right. So two ways to ensure that that's happening. So when we think about all of this, all right, really we are thinking about some cloning, all right, but not cloning like maybe you think about making Dolly the sheep, or maybe trying to make a copy of yourself, which I would recommend you don't want to do. Probably not. All right. Cloning with bacteria is this stuff. All right. Taking a gene out of something and putting into something else and making that recombinant. All right. So as we look at this, and there's a lot of details in here, I've already described most of it as we've gone through here. Our green guy down here is the ampicillin resistance gene. So if we grow it in the presence of ampicillin, if this is in there in our cell, it will grow. All right. I pointed at this one. It's not in there, right? No plasmid here. We have a plasmid, and we've got another plasmid over here. All right. But this one didn't get it inserted properly. This is the right one. All right. So there's multiple potentials that could happen. These guys aren't going to grow on the media because it's got antibiotics in there. All right. Unless they were already resistant, then you'd need to be doing something different, a different plasmid with a different antibiotic resistance gene. All right. So this one's not resistant to it. It won't grow on the plate. Both of these got the plasmid in there, but only this one got it in the right, got the gene where we wanted it, the red guy up above. All right. And this one not. So we can see down below, this is a Petri dish. All right. It's got the antibiotics in there, and it's also got lactose incorporated into the media. All right. So there are color differences, and it's not the same as like looking at a McConkey's plate and seeing pinky coli or something like that, but it's a similar idea. All right. So the blue colonies got plasmids in there, obviously, because they're growing, but they're not recombinant. all right? They didn't get that gene in there. They're not able to use the lactose. And the white colonies indicate that we are, all right? So lactose is being utilized. The white colonies stick out. I feel like the blue ones should be the ones sticking out. The white colonies stick out and let you know all of these white ones in here are the ones that got the plasmid with the proper insert, and it's all growing. So you can screen for it. Now you know, I got my gene, I got it into the plasmid, I got it into the bacteria, it's growing on this media. Every one of these white colonies has that recombinant plasmid. It's a recombinant bacteria. I could then take my loop and scrape a little of that white one off and put it into a gigantic flask, a bioreactor, and I could grow mass quantities of this bacteria, constantly feeding, constantly removing the waste, and the waste harbors my product, right? As it's secreting that stuff out. So we started with the insulin. We would have a vat of bacteria that's churning out insulin, all right? And we would be able to harvest it really pretty easily, all right? That's the idea. And basically all sorts of things. We've got the opportunity to think about getting that gene inserted, and there you go, all right? As long as everything matches up like those puzzle pieces should, you've got it all like it should be, and it grows, all right? So there are other ideas with this, and it's basically what we just looked at, but now instead of thinking about just a single gene, maybe, and I feel like this is probably a type of parasite, looks like a little worm to me. I didn't read through the specifics with this to see if it actually told me who this is, but it looks kind of like a worm. So maybe we're curious not about making one gene, but we're curious about this organism and what genes does it have. So a genomic library basically takes all of the DNA and we cut it into bits, and we take each of those bits and insert them into different plasmids. So you see here, where for simplification, we've got 3 different fragments, all right? 3 different fragments, each inserted into different plasmids. Each of these inserted into different bacteria. We grow them up. We do the exact same thing we saw on the previous slide and verify we got them in there because we get our white colored colonies. And now we've got all of these different bacteria. We could pick them off and cultivate them. We could put them into tubes and save them, put them in the freezer, whatever it might be. But we've got a library where each of these different colored plasmids in here are those respective genes from that organism. And you could have a boatload of bacteria harboring all of that. And then the thought is, what are these products that are going to be generated from these genes we've inserted? And you could do more research into the organism without ever having to, you deal with it once, but you don't have to deal with this. We just look at the products that are being generated from growing up the bacteria, which typically grow considerably faster and in much more amounts from this than from a single organism over here, right? Get a lot of data real quick. So plasmids are probably the most common way when we think about doing some of this genetic engineering and manipulating them. But we can also think about transduction, where a virus is going to infect that bacteria. But in this case, instead of infecting it with phage DNA, we've got host stuff. We've got the genes of interest that we're looking for, all right? And so they could get packaged up, these colored ones up here, could get packaged up into the capsids of our bacteriophage. The bacteriophage could be placed with bacterial cells and give it a little bit of time to try to infect, if you will. And it's basically the same idea at that point. If these phage infect the bacteria, those bacteria become recombinants because now they've got this gene of interest inserted into them. Yes. So in these instances with what we're looking at, these phage, typically their genome is all fragmented up. So they aren't bringing in like all of the viral information needed to do that. So basically, yeah, we're taking all of those properties out of them. And now this is like a syringe, basically, that's just going to be bringing this in and inoculating it into the cell. So that's the idea. And as they grow up, the plaques on the lawn, so all of this bacteria in here, these are where these virus got in there and did it. All right. There's still bits of it that are still functional with some of these little pieces. So we can see phages with the specific stuff that got in there. But the bacteria ultimately harboring this stuff, and the virus was the mechanism to get it in there. All right. So this, I feel like, should have been right at the very start. All right. This is where we take eukaryotic stuff and use reverse transcriptase to make that complementary DNA, the cDNA. So RNA, the messenger RNA, after it's been processed, all right, reverse transcriptase makes the DNA, and then in this case, putting it into the plasmids, and there you go. This is truly not with our worm here, but the very first slide, thinking human insulin, this is the process that would have been utilized to generate that, all right? So they get inserted, and there you go. Now we've got this cDNA incorporated in there. It's another clone library, a cDNA library, all right? Because it's coming from the message instead of straight out of the chromosome. So to get stuff into cells typically can be rather difficult. Some cells are naturally what we call competent. So they are able to pick up free DNA or maybe to pull in a plasmid. They may do it just normally, right? But lots of times cells aren't able to just readily pull stuff in. And so we have ways to try to make them competent, try to sort of weaken the cell wall of that bacteria to allow access to the inside. Sometimes growing them with like some calcium salts helps to weaken that and make cells that aren't competent to make them competent. And another technique is electroporation. So we're seeing this, and this is a technique maybe utilized with eukaryotic cells, as we're seeing right here. All of our recombinant DNA outside of the cell, we want to get it in. So applying a bit of electrical charge, some pulses, causes the membrane to kind of weaken, and it allows these molecules, the recombinant, into the cell. All right. And after after the pulse of electricity, the membrane comes back together, and now we've got some of these things added to the inside. Other ways to manipulate, and I think this is really pretty cool. So we're looking at a cell. We're looking at a part of a cell. Here is the nucleus, all right? And we've got some nuclear membrane around it. Or maybe this whole thing is the cell, and right there is the nucleus. That's maybe more like it. So we're looking at this through a microscope. There is a needle, a micro injection needle, very, very small, very thin, all right, to penetrate into the cell and into the nucleus to put that material in there, all right? And again, eukaryotic stuff, all right? This micro injection needle maybe you have seen like some pasture pipettes, transfer pipettes. You've probably seen the little plastic jobbies with the bulb at the top that will suck some stuff up. But there are glass ones that have a pretty thin piece at the end that we could heat up in a Bunsen burner and stretch out till you almost can't even see it. It's like Louis Pasteur making that swan neck flask, right? Same sort of an idea, but stretching it out so that it becomes super thin and it's still a tube, a needle at this point, piercing into that cell and depositing it into the nucleus. Awesome stuff. All right. So I don't know for sure, but I think with like Dolly the sheep, when she was cloned, I think it was kind of a process like this. And they took a surrogate cell, I believe it was a mammary cell, so a diploid cell, and they took that nucleus and sucked it out, right? So now it doesn't have any instructions. And then they took an egg that had the information that we wanted, and it is going to be what we're going to manipulate. It's only haploid, right? So that diploid nucleus that was sucked out of that mammary cell, it's got both copies. It's got everything we need. And with our little needle, we poke into that cell, inject it in there, and another little jolt of energy, the spark of life, stimulates cell division. And now this egg cell is like, sweet, just fertilized, but it wasn't. And it starts replicating. And now here's Dolly, all right, born from the nucleus of a mammary cell, all right. Awesome stuff. And pretty much this technique utilized to do that. Seems like a simple deal, all right, but not, all right. It is difficult, right? Maybe, thinking having kids, typically, maybe fortunately, that doesn't happen the first time you have sex, although it could, right? Sometimes it takes a bit of practice, right? And then it takes. Dolly the sheep, I think, was attempt 260 something before it actually worked, but they kept trying, right? Because dang it, we want a baby, right? And there you go. This one, I'm just like, what? Some guy had to come up with this. Can't we use a gun, right? Couldn't we shoot it in there? So this is a gene gun. It looks kind of different, but it still looks like a gun. So heavy metal particles get coated with the recombinant DNA, and then we're going to blast it into a cell. And we can do this with some plant cells. This is more common to use a gene gun with plant cells. So a little pulse of helium shoots these particles and blasts through the cell wall of the plant and injects it into the cell. And now we've got a transgenic plant. There's some other ways, I think better ways, although the gene gun's kind of cool, to manipulate plants. So this is a way to do it with the help of some bacteria, right? There is a type of bacteria called Argobacterium tumeficans right here. And these guys are bad news to a plant, all right? If they infect a plant, it causes disease in the plant. It causes some crown gall, causes these big tumors to form like on the trunk of the tree or the stems, whatever, all right? Causes issues for sure, all right? You don't want that stuff, all right? There is a plasmid in this bacteria that helps this to happen, all right? So we would like to utilize that to our advantage, but we don't want to cause disease in the plant, all right? So we manipulate it so that the genes for the disease are not there, but the genes to get you into a plant cell are there. And we manipulate that plasmid to put the gene of interest that we wanted, and we expose it to the plant, all right? Typically like individual cells, and it's a slurry of stuff, and then you grow them up. Here it just looks like, here it is, right? So the DNA that we want within this plasmid, it gets incorporated and then inoculated with these cells. It brings it into the cell, gets into the nucleus, and now you've got a transgenic plant growing different stuff, all right? Whatever it could be. So we have the ability to manipulate bacterial cells, the ability to manipulate eukaryotic cells, animal or plant cells. All of the genetic sequence is the same. There are introns that we see in the eukaryotes that require some processing if we're going from a eukaryote to a prokaryote. But if it's just eukaryote to eukaryote, animal into a plant or something like that, you don't need any of those other processing steps, all right? And it gets inserted directly. Pretty cool stuff, all right? And I'm thinking with this, where we're seeing our bacterial cell, there's still got to be some processing if we're thinking eukaryote into this prokaryote, but the other stuff, not so much. So all of those different ways to get recombinant stuff, all right? Now we want to think about some like detection stuff, playing detective CSI sort of stuff, all right? So here we've got a sample of DNA, and maybe we've, extracted some body fluid, and we're thinking some sort of disease, all right? So there could be all sorts of DNA within there, all right? We denature it. So like PCR reaction, we use a little bit of heat to cause the double-stranded molecule to break apart. Once it's unzipped like this, now we've got some DNA probes. And these probes have some sort of tag on them that will allow us to know if it's linked up or not. So maybe let's flip it back to that first image where we saw the chlamydia, all right? Maybe that's what we're curious about. And we've taken some secretions. We have put them into this situation where the DNA gets unzipped. And now I've got a little DNA probe that is specific for chlamydia. And if chlamydia was in that sample, when I unzip its DNA, my probe is going to link up to it. If chlamydia is not there, my probe's not going to do anything, right? And if it does, it binds and we can detect it, all right? And then we know it happened. It linked up and we've got a positive. Nothing links up, negative, all right? Pretty cool. So another technique looking at DNA, agarose gel electrophoresis is utilized for examining DNA molecules. Oftentimes when we think about this, restriction enzymes are used and we make some cuts. So we get individual pieces instead of just trying to look at an intact chromosome. That's not going to be very informative. We want to see fragments. If you've got an entire chromosome, maybe from us, we expose it to these enzymes and even just one chromosome, you would assume that these enzymes are probably going to make multiple cuts in that chromosome because there's probably those specific sequences repeated a few times in that. So I might get 3 cuts in this chromosome. And maybe I take one of your DNA and I expose it to the same thing, and maybe it gives me 4 cuts or 2 cuts. And we could run all of our stuff out on a gel and get a fingerprint, if you will, that shows some of this variation. And that's what we're looking at down here. The ladders, these are fragments of known sizes of DNA, controls, if you will, that have been added to the flanking regions. And all of the stuff in the middle are individual samples, all right? The inside stuff has been cut with these enzymes. And we can see some similarities and some differences as we move through there. This is an idea for stuff like paternity testing, where you could take a couple of different male samples expose them to these enzymes, run it out, take the mother's, take the child's, and run them out as well. The mom is going to be contributing half, the dad will be contributing half, and the kids got one of each, right? So looking at all of these things, you should be able to figure out some of those details, all right? So it's exposed to some current. You can see over here, this would be this chamber. That's what they're loading up right here. Up at the top is negative, down at the bottom is positive. DNA is negatively charged, so it wants to move towards the positive pole, and it starts going through this gel. And it's like a sieve. So bigger things get kind of stuck and they stay more toward the top, and littler pieces move quicker and move toward the bottom. And that's what our ladder is showing right here, all right? Bigger stuff and littler stuff, and then our stuff's down here toward the tail end, all right? That's the idea. So using those enzymes, it makes those cuts, and we can see these differences. RIFLEP is restriction fragment length polymorphisms. So cutting with that known enzyme and seeing those patterns. But it's important to think about, like paternity testing, making these cuts with the same thing, right? And then you can see, and in this case, looking at normal versus mutated, there's some similarity and then there's a few differences, right? Where maybe this one that's one big fragment, maybe it got mutated and now it's like a dimer. It's two smaller pieces, potentially, right? Little bit of variation with that, showing different things occurring in this situation, normal versus mutant. So we can run it out on the gel. We can see those banding patterns. Oftentimes the DNA, when we're running an agarose gel, we use ethidium bromide. And ethidium bromide, that's what we're looking at over here, when it's actually ran out, and then we take this photograph, the ethidium bromide is going to light up, all right? And we can see it where it's linked up into the DNA. It's one of those intercalating agents, right? And so as it lights up in here, and again, moving through, you can photograph it and ultimately make some measurements and determinations, right? And who knows what this one's actually looking at, but samples. So then we could take it a step further and do some blotting. And there's a couple of different blots. Southern blotting works with DNA. Western blotting works with proteins, and northern blots work with RNA, all right? There is no Eastern blot. Darn it, all right? So, 3 different types of blotting. And ultimately what you do, and in this case it's DNA, the DNA has been run out on that gel, and then we want to transfer out of the gel. We basically want to suck the DNA out of there and put it onto a membrane. That's what's happening. So it's a sandwich that gets created. You've got this buffer, you've got a piece of paper, filter paper, you've got your agarose gel, then you've got the stuff you want to make the transfer to. This is a membrane. It's where we want to stick the DNA onto it. There should be another piece of filter paper up here, paper towels, whatever, and then a weight to keep it all kind of sandwiched together. You expose that to current, but now instead of trying to get it to go down, we want it to get it to just move over, right? So it's different, pulling it to that membrane. And then you can see, as it's lighting up, specific pieces, fragments of those, of the DNA that are lighting up on that membrane, all right? And these that are lighting up because of this. We've added some probes to that mixture to try to get it to bind. If it binds, it's telling us details about those specific fragments, all right? That's what we're seeing right here, all right? To allow it to light up. In this instance, this is Same but different. This is a microarray. So this is a big slide, bigger. It's different than the microscope slides we think about. All right. And also super expensive. All right. Handful of years back, these were like thousands of dollars for an individual slide. I'm assuming now they're a little bit cheaper, but it's still costly. All right. These slides have all of this genetic information impregnated onto the slide. All right. So an entire genome of somebody, right? A bacterium, a fish, whatever, right? We've got something on there. And then we can probe it, if you will, with specific things. And here we're looking at cancerous and healthy cells, making some complementary DNA, and then hybridizing them, trying to get them to go onto this array. If they stick to those specific regions, we use some lasers to read it. You get this, I'd like to say checkerboard pattern, but it's not a checkerboard. It's all sorts of craziness. Greens and reds, different intensities of this indicate what's being expressed. So the green only in healthy, red only in cancerous, and yellow, which I see just a bit of it, are expressed in both. So you can see what's happening with this and get kind of an idea about it. And knowing, and you can't see any of this, and I probably in the textbook, you can't see it either. Each of these lines and all of this over here, we've got rows and columns that are different parts, different genes, chromosomes, and we can figure out where these things are linking up, all right? Pretty interesting. So proteins, we can evaluate proteins. There's a couple of different ways that we think about with this, but running them out in gels. SDS page, this is a denaturing gel. It's got SDS, which is sodium dodecyl sulfate. There's a couple of other reagents that get added to basically denature that protein to cause it to unfold. And then we run it out in the gel, all right? And we can see over here the bands that are showing up, all right, as the proteins start migrating through that gel. There are other types that we call native. You don't put those detergents in there and they stay intact. Maybe if we want some blotting, we want to keep them intact. So we would run a native gel instead of a denaturing gel. And then, well, that's PCR. Like that southern blot, we can make a sandwich and we can transfer from the gel to the membrane and we can do probing with that, with different antibodies linking up to specific protein fragments and lighting up or not, all right, to get details. This is just the proteins that have been migrated, not that. PCR, we already talked about, multiple cycles, heating and cooling to get mass quantities. And the way we can think about for detecting some of this stuff, there are sequencing reactions more than just making the stuff. We want to know what's up, all right? So a dideoxynucleotide is similar to our nucleotides, deoxynucleotide, but instead of it lacking only one, it lacks two oxygens, dideoxy, all right? And these guys are odd because when they get incorporated into the growing strand, it makes it stop growing, all right? They also have some sort of a tag, fluorescent tag probably linked up to them. But as soon as this one gets incorporated, it's a done deal. PCR reactions are just churning out multiple rounds of replication. So the thought is, if you were to add some of this, the very first base will wind up with one, the very last base, and everybody in between, after hundreds of rounds of replication, you're going to get all sorts of little pieces. And you run those pieces through a machine that counts, and it knows X amount of bases. Here's my one. I was 5 in, sixth one is this, seventh, eighth, ninth, whatever it may be. And they show up. You get a computer printout done deal. All right. Sequencing. So we've replicated, and now we know what it is that we actually replicated. And then we can do some more stuff with it, bioinformatics, and try to understand what was all of this actually code for. This is the same sort of an idea, but it's in a capillary gel. So you've got all of these fragments in a gel, and they're basically being eluted. They're moving through this gel, and as it gets through to the laser, that's where we do the detection. And in the computer, it's showing us all of these little spikes. And it's just this continuous thing, dripping it through, reading every single base as it's going through. All right. Pretty cool. And then this text stuff in the book talking about, I want to say it was Clostridium, but I could be wrong. But same Riflip stuff, looking at Javier's strain that was isolated and this strain that it matches up with. That's the idea. Or maybe it was C. diff. Maybe that's what we're looking at. So that's the idea. And I know we're running out of time. I'm so close to the end, I'm pretty sure. Another thing that we could think about are some fluorescent proteins that can sometimes be used as markers to show that stuff got in there instead of the antibiotic resistance. I feel a little disturbed with these mice. I feel like that's almost nightmare worthy, with the glowing green eyes, nose, tail, and even ears. All right. We've got some of them in some bacteria, and then screening for some of that with antibiotics in those plates. That's the idea. So there are other ideas to stop some of this, and cells, eukaryotic cells, can make some antisense RNA that link up to message to shut it off. And that might be important when we think about virus trying to get in, and we want to shut that down, blocking translation, blocking the products, and other mechanisms that also do the same thing. Small interfering or microRNAs that shut this stuff down. Lastly, gene therapy. We think about therapy, not cures. There have been a few individuals that have actually been cured, but there's also been some that have been killed. Killing people is not what we want to do in medicine, right? We want to help. And so we now think more like therapy. We can take cells out of a person. We can incorporate a good gene into them, grow those cells up, and pipe it back into the people. Those cells, short-lived, maybe half a year, maybe a little less than that. So you have to get multiple treatments, but you feel better, right? We have tried to manipulate stem cells and get stuff put in there, and we've had some success, but we've also had some problems. So we'll think a little bit about some of that stuff a little later when we talk about some infectious disease. SCID, severe combined immunodeficiency, was one of the first ones that some of this was worked with. Maybe you've heard of David the Bubble Boy. He wasn't helped, but some people shortly after were. All right. Thank you. Good day, good weekend.